Hi, my name's Rebecca Cockerham, and we're going to talk polymerase chain reactions. First step is you take a buffer and a digestive enzyme mixture. You place said mixture in each aliquot tube with samples. Place those samples on the heat block and let them heat for about 2.5 hours at 56 degrees Celsius. From there, you spin down on the centrifuge for a few seconds. Following that, you add a 5 mole sodium chloride solution, following with 100% ethanol. You give it a little shaky, and then you place them in the centrifuge again for about 20 minutes. So now you extract the liquid from each aliquot and replace it with a 70% ethanol solution. Then you once again place them in the centrifuge and spin them down for. Alright, and following that 10 minutes spin on the centrifuge, you remove your liquid from your aliquot. You allow your samples to air dry, but not too much. And then you add H2O. And then you store your samples at 4 degrees Celsius and chill out. Alright, and to prepare your samples for the thermocycler, you make a master mix of forward and reverse primers, DNTP, buffer, H2O, and Taq polymerase. You put a portion of your master mix in each aliquot in the tray, and then you add a small touch of your DNA sample. You place your tray of samples on the thermocycler and you let it work its magic. And while you're waiting on your cycler, you prepare your agarose gel with agarose, ethenium bromide, and a 1x TAE solution. So after the completion of the thermocycler, you remove your samples, you add a loading dye to each sample and transfer the samples to the gel, applying 1 sample per well. And finally, you image your gel and analyze your results based on viewing the banding pattern with the ladder that is to the left of the gel. And hopefully, if your experiment was successful and you did everything you were supposed to do, you were able to target your gene in question.